This video gives an introduction into the bioluminescence phenotyping system PhenoAIxpert ES by LemnaTec. It shows how to operate it, and in the end there is also an example of an overlay of an RGB image and a bioluminescence image.
Hello phenotypers! Let's have a look at the LemnaTec PhenoAIxpert ES. The PhenoAIxpert ES is a system that has an enhanced sensitivity camera. Why using an enhanced sensitivity camera? This system is dedicated to bioluminescence. Bioluminescence is a function that is used in research for labeling metabolites or microbes. And imaging bioluminescence can serve to monitor where the reporters, so the luminescence, are present at what time. So it tells you about where and when, for instance, the metabolites or the microbes are present and active in your sample. Classically, luminescence is used in microscopy. However, here, with the PhenoAIxpert ES at LemnaTec, we have the possibility to monitor bioluminescence at whole plant level combined with classical phenotyping. For monitoring bioluminescence, we use the PhenoAIxpert ES. It is the cabinet here behind me. It is a phenotyping cabinet that is light-tight because when you monitor bioluminescence, you have to exclude any external light. This is due to the high sensitivity camera that images very weak light signals. And you can imagine if external light comes into the box, then the imaging of luminescence is overrun by the external light. So therefore we need a light-tight cabinet. The system also has a computer workstation and it has a chiller. The chiller serves to cool down the camera. This is necessary because the high sensitivity camera only works when the chip is cooled. For working with the PhenoAIxpert ES, we need to switch on the system as such, have the computer running, and we need to switch on the cooler. When everything is running, we can use the computer workstation to access the functions. This works via the LemnaTec control software that is installed on the computer. When the system and the software are up and running, we can use the system for recording images. As mentioned, it can do classical phenotyping, but it can be also used for monitoring bioluminescence at whole plant level. Let me give an example. I have here a tray with some tobacco plants. We will use the tray to take a classical RGB image of the plants, and we will check whether in one of the plants is a luminescent signal that might come from a labeling of metabolites or microbes that could have infected the plants. So to do imaging, of course we need to open the cabinet. And when the cabinet is open, we can insert the sample tray. So now the sample tray is on the sample stage below the cameras, and after removing the protective caps, we can access the cameras in the software to record images. Having placed the sample inside the imaging cabinet, the plants are now located below the cameras and the lamps that are required for RGB imaging. The luminescence imaging with the high sensitivity camera of course does not require illumination because there the light is directly emitted from the sample. To record luminescence, of course we need to close again the door. As mentioned, bioluminescence can only be imaged when it is completely dark and external light is excluded. Now the samples are in the cabinet and we can use the cameras for image recording.
In the Lemna control software, we can do all settings that are required for the RGB camera and for the high sensitivity camera that is required to image the luminescence. Here we also give in experiment and sample name and trigger the image recording. Then the system takes images. Here is an RGB image, so a classical photograph of the samples. And this is the luminescence image. We see some bright spots. These spots indicate where in the samples luminescence could be found. When laying the luminescence signal over the photograph, we see that the luminescence appears in two plants. So for the example, this means that these two plants exhibit the luminescence signal.
When image recording is finished, we can take out the samples and now the system is ready for the next recording. So we could insert a new sample and start the recording process again. The images that have been recorded with the system can be processed in the LemnaTec software. Thereby, image analysis can be used to do classical phenotyping, so measuring sizes of the plants, and it can be used to analyze the luminescence.
For more details on PhenoAIxpert ES, visit our webpage and contact us directly.